In this video, we will look the structure and different types of the antibodies, that what are their functions. So let's begin the video. Guys, moving to the next slide, we'll continue the video. So next slide is on our screen. You can see here. The first question is, what is antibody? So we must know what is antibody as we are going to discuss about it. The diagram appears here on the screen. Is shown antibody. Mm, Antibodies are the globular protein which belongs to the immunoglobulin family, that is nothing about Ig. All the antibodies having similar structure, as you can see here on the screen. There are various regions inside the antibody. You can see here where the cursor it is antigen binding site, where the antigen comes and binds to it and then it is destroyed. So this is also known as the light chain, and this one is also known as the heavy chain. The light chain and heavy chains combines to each other and makes the Y shape, that is our antibody. And guys, this both the light and the heavy chains are bounded to each other with the help of sulfide bond, that is disulfide bond to each other. That is also known as sulfide bridge. It is a kind of covalent interaction in between both these chains. And here you can see it is written here variable constant. That means these regions are not constant, they are variable. They keeps changing on the basis of the requirement of the antigen, that what kind of the antigen is going to bind with it. These antibodies need to adjust themselves according to the antigens. Guys, this both the light chains of the antibody is made up of total 220 amino acids. That means this each of the light chain is bounded is made with 100 to 110 antibody and amino acids, and the heavy chain is consist of 330 amino acids. Now, if we talk about the types of antibody, then usually antibodies are of five types. They are classified on the basis of their structure, weight, and the functions. So we'll discuss about each antibodies one by one. So moving to the next slide. As I have told that antibodies are classified into the five types. You can see here the first one is IgG, IgA, IgD, IgE, IgM. So these are the various types of the antibodies, classification of the antibodies. There is a very simple word to remember all these antibodies at the same time, that is GAMED, GAMED, that is G for IgG, M for IgM, and A for IgA, and E for IgE, T for IgD. So this is a kind of simple word by which we can remember all these five antibodies at the same time. So we'll start discussing about the IgM. So for IgM, we'll have to go to the next slide. So here the structure of the IgM is appearing at our screen. You can see the structure of the IgM antibody, and here it is giving the size, that is nothing about the weight of the IgM antibody. The weight is 970 kilodalton. Seeing immunoglobulin structure, it is pentameric in the structure. Guys, pentameric means this five sites, which this antibody is having five sites. This is means the pentameric, and the location that where this antibody is found inside the human body, that is lymph and the bloods. So this antibody IgM is found usually inside the lymph and the blood of the human body. And the binding sites, if we talk about binding site, that is what is the binding site here. So you can see it, this is the binding site of this antibody. Binding siting do nothing but it binds to the antigen and it destroys the antigen. After binding the antigen to the binding site, it goes to the this crystalline region, fragment of crystallization. This is also known as the fragment of crystallization, and this after the neutralizes the infection, the virus, whatever antigen bonded with this. If we talk about the concentration of this IgM inside the human body, then it is usually 5 to 10%. It is found in, you can say it is found in a low amount, but this is not so low. The functions of the IgM antibody is it is the first line defense of the human immunological response system. It binds to the antigen and activate the complement system of the antibody, and further it destroys to the antigen and remove the waste out of the body. So going to the next part of the antibody. So next we have IgG antibody. Is its structure is very simple, as you can see here on the screen. FC, that is fragment of crystallization, and this is also known as the variable region, and this is a constant region. This one is the constant and this one is the variable. And here it is also given the light chain and the heavy chain. If we talk about the concentration of the IgG antibody inside the human serum, that is 70 to 80%, that is very high. And the weight of the IgG antibody is 150 to 150 kilodalton actually. And its type, it's alpha type, it can cross the placenta. That means it's a only kind of antibody that can cross the placenta and provide the immunity to the fetus present inside the mother. It provides the immunity to the fetus just after crossing the placenta of the mother, because the fetus is not having any kind of his own immunological response to protect his body. So that is why this IgG protects the fetus. If we see, the IgG is having four classes. The one is Ig1, Ig2, Ig3 and Ig4. It is further divided into the four classes which have their own different work, but these all four are interdependent to each other. They regulate to the each other in protecting us from the antigens. If we talk about the lifespan of the IgG antibody, it is usually 23 days. Sometimes it varies 21, 22, 23. But if we're gonna talk about the average, then it is 23 days. It also helps in the bacteria immobilization. That means it will bind to the bacteria and activate the complement system, after which the immune system of the human body will destroy that bacteria via different processes. So this was all about the IgG antibody. So moving next to the next antibody, that we have IgA. As you can see here on the screen, this is the structure of IgA antibody. The structure of the IgA antibody is a little bit different from all other antibodies, as you can see here. In case of IgA antibody, you can see here, these are the FAB region, that is a fragment of antibody antigen binding sites, and these are the FC region, that is fragment of crystallization region. Now you can see here, these both the regions are responsible for the binding of the antigen to the antibody. And the weight of the IgA antibody is higher. Obviously it will be higher because it is having many different components in it, that is 320 kilodalton. And concentration, that is 10 to 15%. It is found 10 to 15% in the human serum, and capable of transcytosis. Transcytosis is nothing but the process of crossing the epithelial layer of the skin. This IgA antibody body is also found inside the saliva, tears, and the gastric secretion in the breast milk. Um, in the breast milk, this IgA helps to fight with the infection from the infant, as the infant takes this milk from the mother. So this milk develops a different kind of the immunological response inside the baby's body, and this antibody kills the different viruses, bacteria which are present inside the gastrointestinal tracts and the respiratory tracts as well. The last point you can see here, provides immunity to the infant via breastfeeding, as I have already discussed, that when the infant takes the milk inside its body, then its body develop a different kind of immunological response to the foreign antigens. So guys, this was all about the IgA. So moving to the next antibody we have, that is IgD. IgD is here on our screen. Its structure is also very simple as like the other antibodies. It also have just like FC region and FAB region. If we talk about the weight of the IgD, then it is 180 kilodalton and it is type of a delta heavy chain. And the concentration, that is the amount of IgD inside the human serum, that is less than 1%. It is present in very low quantity inside the human serum. This IgD antibody expressed with the mature B cells. Actually, the function of IgD body is not defined yet, so the function is undefined. So it expressed with some kind of another responses, that is B cell, that is present inside, which helps us to find with the pathogens to protect our body. So this was all about the IgD. Now we will be discussing about the next antibody, that we have IgE antibody. You can see here on the screen the structure of the IgE antibody is also very similar to the other antibodies that we have already discussed. So their FC and FEV are similar, the function is also similar, the bonds present between them is also same as like that. There is no difference inside the structure of them, but there's a slight difference in the function and the weight and the concentration, that is the amount at what it is present inside our serum. That the weight, if we talk about the weight, that is 200 kilodalton, and the concentration is 0.3%, that is very very less. Function is mediating allergy reactions. It usually recognizes the allergic antigens and after recognizing binding to them and destroy them. IgE also shows the immediate hypersensitivity reactions with the symptoms such as fever, sneezing, asthma, shocks, etc. These are the symptoms which appears on behalf of the hypersensitivity reactions which are being carried inside our body. If there is some kind of error in these reactions inside our body, then obviously the IgE will show the response by giving some unique features to us, that is sneezing, coughing, fever, or the shock. So guys, this was all about the antibodies. We have discussed about the 5 antibodies. So make sure to subscribe the channel. Don't forget to like the video, comment on the video, and to share the video. So that's it for this video. Now we'll see you in another video. Thank you for watching this video.